Hello everyone, I am Neelima. You all know me. I am currently doing PhD in IIC Bangalore. So today I want to teach you one technique which now I am using my PhD program. So this is in vitro transcription. We all know about transcription. So it is the production of RNA from DNA. Now in all kind of cell like prokaryotic as well as eukaryotic, we can see this process. We can't see this process, but this process is happening continuously, so that a particular amount of RNA can be maintained in the cell. So now after the formation of RNA, the protein production will take place from RNA to protein. It's called translation. But today we will talk about in vitro transcription. So whenever it's inside the cell, this is called in vivo process. But whenever it's outside the cell, it's called in vitro process, right? So in vitro transcription process actually takes place within the append drop. That's why it's called in vitro transcription, because it's so we actually purify all the components required for this process, and in the append drop we added all the components required for in vitro transcription so that it can happen. And at last we'll check the RNA production, because transcription means the production of RNA. So at last we will check the RNA production, the amount of RNA. So now for detection of RNA production, like for detection of the amount of the RNA production, we can that we can do in different process. Like we can do through gel-based process, like if you use radio-labeled RNTPs, because for the production of RNA we have to add the RNTPs. So if you add radio-labeled RNTPs, then you can detect that through the gel some radioactive assays. But in my case, I am using something called ribogreen. So ribogreen is nothing but it's actually the combination of two ETBR. ETBR, you all know, it actually the intercalating agent which actually intercalate in between the base pairs of the DNA. So ribogreen is like the same, but it's also intercalating agent only, and it can bind both in DNA and RNA. So to remove all the DNA from the reaction mixer, because it's transcription process, right? So for the transcription we need the DNA promoter, the DNA template. So after the transcription process, we will treat the complete sample with DNAs, so that our DNA will be not there after the treatment of that reaction mixture with DNAs, and then we will check through the ribogreenation. So I'll be discussing all the things, all the required steps, the component needed for the assay. So stay tuned. So now first we will discuss the IVT buffer component. So IVT buffer is something called in vitro transcription buffer. So whatever normally present inside the cell, we try to mimic that condition. So because of that, we make this IVT buffer and use in our assay. So for this IVT buffer, we have something called Trees HCl, which maintain the pH 7.5, because inside the cell we know the pH is something 7.5 like that. So we try to maintain that pH. So we have added trees for maintaining that pH. The working concentration in the solution is 50 millimolar. Other than that, we need KCl, DTT, MnCl2, MgCl2, glycerol, and water. So the water is something called DEPC treated, so that so because of the DEPC treatment, RNase will be not there. So if RNase is there, then it's a problem, because it will degrade your RNA. The whatever amount of RNA is producing through the reaction, this if you add the DBC treated water, then RNAs will be not there. It can't clip the RNA. So you can try to Google it to see the structure of DBC as well as how it actually works in the solution, but you have to add DBC treated water only. And I have mentioned all the working concentration of those compounds. You can use, if you are doing in vitro transcription, you can maintain these concentration of all the compounds here. Now for the transcription process, we need some compounds like DNA, which is the DNA template. So actually I am working on Mycobacterium tuberculosis, so I'll specifically use something called AP3 promoter, which actually like scientists are using this promoter for Mycobacterium tuberculosis. Other than that, we need RNA polymerase, that is the main compound, and sigma protein, that is the sigma factor. Other than that, MTB use two other protein molecule, which is named as CARD and RBPA. So this is particularly for Mycobacterium tuberculosis. These are the essential protein for Mycobacterium tuberculosis, because recently one group called Jensen et al., they have proved that based on the presence of CARD and RBPA, the transcription can, like, based on the concentration of the CARD and RBPA, transcription its rate actually regulates by these two proteins. So you, for the at least MTB, because it's essential for MTB, these two protein components are required. And I have mentioned all the concentration needed for these assay of these essential compounds. Now I'll talk about the protocol for the in vitro transcription process. So first, what we do, we generally first make the IVT buffer. So whatever I told here, that the components of needed for the IVT buffer and all the concentration. So accordingly, we'll first make the IVT buffer, and with the IVT buffer we will add the RNAP and sigma factor, because for RNAP to sit on the promoter, it needs sigma factor. So we will first make the holoenzyme complex. After adding RNAP and sigma factor, it will interact and produce a holoenzyme complex. So and if you want to see the effect of any inhibitor which actually inhibit the binding of RNAP and sigma factor, you can add in this step. This inhibitor you can add here. After that, what we will do for the formation of transcription, like it's not with promoter now, it's only RNA polymerase and sigma factor. To make this proper enzyme complex, we will incubate it at 37 degrees Celsius for 10 minutes. After that, we will add the DNA, the promoter, whatever I am working on AP3 promoter, so I'll be adding the DNA after that. After the adding of DNA, we will again incubate it at 37 degrees Celsius for the 10 minutes. And if you need to add any essential protein, that is CARD and RBPA for MTB, that you can add after the adding of the promoter DNA. So next we will add CARD and RBPA and again incubate it for at 37 degrees Celsius for 10 minutes. Now after the adding all the components, now we need to start the reaction, because I have the RNA polymerase, I have the sigma factor, I have the promoter, all the things are like I have added. Now we need to start the experiment, need to start the process. So to begin the process, we need to add RNTPs, right? Because RNTPs are the main component which will start the reaction, because one by one the RNTPs will add and the total stretch of RNA will produce. So for that, next we will add the RNTPs. So whenever we are adding the RNTPs, after that we will incubate the a reaction mixer for 1 hour at 37 degrees Celsius, and this is the time where actually RNA will form, because we have added all the things. Now we are adding the RNTPs. So after adding RNTPs, we will incubate it for 1 hour 37 degrees Celsius. Now what we will do, after production of the RNTPs, RNA will produce, right? But in the reaction mixer, we also have the DNA as well. But if we add any intercalating molecule in this step, it can intercalate in DNA as well. So it will give us wrong, like false positive or false like measurement. Because see, let's say you have 10 molecule of RNA has produced in this reaction mixer, but you have 10 molecule of template as well. So if you add any intercalating reagent here, it will intercalate in the RNA. I can understand, but DNA is also there, right? So it will intercalate in DNA as well. So now we have to remove the DNA. So for that we will add CaCl2. That is the calcium chloride. The working concentration is 5 millimolar and DNA is 1. So DNA is 1, what it will do? It will go and cleave, chops up all the DNA. So now in the reaction mixer we have only the RNA. So what we will do? So after the adding of DNAs and CaCl2, we have to incubate this reaction mixer at 37 degrees Celsius for 1.5 hours, so that DNase 1 can go and chops up all the DNA present in the reaction mixer. So now if you are doing some radioactivity based assay, you have to load on the gel. This is something, it's very if you are using any radioactive molecule, you need to maintain the process, you need to take care of yourself as well, because these radioactive things are not good for your health. So what I am doing now currently is ribogrin based assay. So what is ribogrin? I just told you, it's an intercalating agent. It will go and bound with the intercalating the RNA, and through the fluorescence count, so it's a fluorescence molecule, we can just take the fluorescence count, so that like based on the fluorescence count we can say like how much RNA is producing. Because let's say in one reaction mixer you have 10 molecule of RNA, but you have added some inhibitor in a different reaction mixer. So now here we have 10 RNA, but where you have added the inhibitor, let's say 2 RNA has produced. Now a ribogrin will go and ribogrin will intercalate in this 10 RNA, right? But here you have only 2 RNA. So ribogrin will go and intercalate in this 2 RNA. But as you have 10 RNA here, so it will show increased fluorescence, more fluorescence than this one. This have only 2. So based on that, we can take the just count, fluorescence count, and we'll be done with it. So based on this, we can say the amount of RNA is producing in the reaction mixer. But one drawback is that you can't say what is the length of the RNA. So in the ribogrin assay, ribogrin based in vitro transcription assay, we can tell that what is the amount of RNA which is producing in the reaction mixer, not the length. Because for the length you need to go through this gel based assay. So this is the in vitro transcription process I have just discussed. So these are the components of the IVT buffer. These are the most important thing, because these are the base of the reaction mixer, because we need to add a DNA, that is the DNA template, RNAP, sigma factor. If any essential molecule, essential protein is there in that organism, you have to add that as well. And yes, this is the protocol of this in vitro transcription process. If you are doing any IVT assay, I'll recommend please go through this ribogrin based assay. You can go to the Thermo website, and you can check this the assay protocol, and all of this they have already given in this in the website as well. So and if you need to check the length of the RNA, then you have to go through this gel based assay, because length you can't detect through the ribogrin assay. So this is all about in vitro transcription. If you have any question, please let me know. I'll try to help you. Thank you. Bye bye.